mutations or knockout of any of these genes happens, this leads to hepcidin deficiency, unregulated ferroportin activity, and iron overload. Now, my entry into the iron field came about because I was studying hemojuvelin for a different reason as a postdoc in Herb Bloom's lab, and we were where we discovered that hemojuvelin is a member of a family of 3 genes called the repulsive guidance molecule, or RGM family, that we showed can function as co-receptors for the bone morphogenetic protein, or BMP signaling pathway. So what are BMPs? They're a subfamily of the TGF-beta superfamily of signaling molecules, which is a large superfamily of about 40 members that includes the TGF-betas and BMPs themselves, as well as growth and differentiation factors, or GDFs, activins, inhibins, and malaria inhibiting substances. These are disulfide link dimeric proteins that share overall structural similarities in a common paradigm of signal transduction, whereby these dimeric ligands will bind to complexes containing 2 type 1 and 2 type 2 serine threonine kinase receptors. Upon formation of the complex, the type 2 receptors will phosphorylate the type 1 receptors, which then phosphorylate intracellular SMAD proteins. There are 2 subsets of SMADs, SMADs 1, 5, and 8, which are activated by the BMP ligands, and SMADs 2 and 3 that are activated by activin and TGF-beta ligands. These SMAD proteins form a complex with a common mediator SMAD4, and the SMAD complex then translocates to the nucleus, where they act as transcription factors to regulate the expression of genes, leading to a diverse array of biological functions. Now, an interesting question in this field, I mentioned there were over 40 ligands, and they lead to a diverse array of biological functions. How is it that this can happen in a cell context specific fashion when you have a very limited subset of only altogether 5 type 2 receptors, 7 type 1 receptors, and these 2 subsets of SMAD proteins? Well, this is a complicated question and answer, but at least one of the mechanisms by which this happens is that there's regulation of this pathway at a number of levels. And one type of regulation is through co-receptors. And this is where our data suggests that the RGM proteins, including hemojuvelin, are functioning as co-receptors for the BMP SMAD signaling pathway. And this is based on data such as the following. So if we take, this is an experiment using liver-derived cells in culture, and we transfect them with a BMP responsive luciferase reporter, where if we treat these cells with exogenous BMP ligands, we get a nice induction of BMP luciferase activity. We see a similar induction in luciferase activity if we transfect in cDNA encoding hemojuvelin, or in fact, any of the RGM proteins. And this is specific for the BMP side of the pathway, because if we use other reporters, for example, the CAGA luciferase reporter, that's a SMAD3 specific reporter that responds, for example, to TGF-beta signals, we see no effect from hemojuvelin transfection. In the data that I don't have time to go into, we showed that that hemojuvelin is working through the canonical BMP SMAD signaling pathway through endogenously produced BMP ligands, BMP receptors, and BMP SMADs, because if we inhibit or knock down any of these individual components, we can block this HGV-mediated signaling. Now, as we were generating these data, Papanickel and colleagues published their landmark paper linking mutations in hemojuvelin to juvenile onset hemochromatosis. So of course, we asked ourselves the obvious question, whether this BMP signaling function of hemojuvelin were important for its role in iron homeostasis regulation. And of course, the role that we thought about was in the regulation of hepcidin, since this is where hemojuvelin had been proposed to act. And indeed, we found that BMPs are quite potent stimulators of hepcidin production. This is an in vitro assay in liver-derived cells where we stimulate them with exogenous TGF-beta superfamily ligands and measure hepcidin expression by qPCR. And you can see that BMPs are quite potent stimulators of hepcidin expression by hundreds to 1,000 fold. And that many BMPs, when you add them exogenously to these cells, can stimulate hepcidin, including BMP2, BMP4, 5, 6, 7, and 9. But it's really the BMP subfamily that are the most potent stimulators of hepcidin compared with other superfamily members. And subsequently, it was shown that this is the BMPs are regulating hepcidin directly at the transcriptional level through specific SMAD binding elements on the hepcidin promoter. Now, subsequent work in the lab has been focused on identifying which of these BMP ligands is important for regulating hepcidin expression endogenously in vivo. So many of these ligands can stimulate hepcidin if we add them to cells in culture, but which ones are important in vivo? And clues for this came from two directions. One is some of our work looking at the binding affinity for the BMP ligands for this co-receptor hemojuvelin. We had published in our original paper that there is a direct binding interaction between BMPs and hemojuvelin. And in fact, the crystal structure of this complex was solved about in 2015 by Christian Siebel's group. And you can see here that hemojuvelin, which is also known as RGMC, binds directly to BMP2 ligand. And interestingly, the location where hemojuvelin interacts with the BMP ligand is actually the same binding site as the type 1 receptors, which are the receptors that actually phosphorylate the SMAD proteins. So it's an interesting question that I think is still really unanswered, is mechanistically how hemojuvelin is functioning to enhance the signaling response in response to BMP ligands. And I think more work is needed in this area to really understand how that is working. But here I'm going to show you some of the data generated from our group, where we used surface plasmon resonance to look at the binding interactions, the binding affinity between the different RGM proteins and the different BMP ligands. And here I want to mention that BMP ligands actually have different subfamilies based on structural similarities and some differences between each other. So BMP2 and 4, which are colored in blue, are part of class 1 BMP ligands. BMP 5, 6, and 7, which are colored red, are class 2 BMP ligands, and BMP 9 is another class. And here I'm focusing on the BMP ligands that we showed could stimulate hepcidin in vitro. And so what we found is that all of the RGMs have a relatively highest affinity for binding the class 1 BMP ligands, BMP 2 and 4. And actually, none of the RGM proteins were able to bind directly to BMP 9. But interestingly, what was unique about hemojuvelin compared to the other RGMs is that it had a relatively higher affinity for the class 2 BMP ligands, BMP 5, 6, 7, compared to the other RGMs. So if we were trying to think of something that hemojuvelin might do that might not be well compensated for by other RGM proteins, we thought it might have something to do with these class 2 BMP ligands. Another clue to understanding which ligands might be important endogenously in vivo came from a screening assay done by Maria Paul Ross group, where they took mice and they put them on a low iron diet, a controlled diet, and a high iron diet. And as you can see here, as we discussed previously, the low iron diet will suppress hepcidin expression, whereas the high iron diet induces hepcidin expression. And so what their group did is they did a microarray analysis of the liver from these mice to ask what other genes are regulated by iron concordantly with hepcidin as a means to discover novel hepcidin regulators. And in their unbiased screen, they determined that BMP6, the ligand BMP6, is actually regulated by dietary iron in the liver. And this has subsequently been confirmed by our group and others. And here I'm showing you the data from our group, where we show, in fact, that a low iron diet decreases BMP6 expression in the liver, whereas a high iron diet increases BMP6 expression in the liver. We subsequently looked at the other BMP ligands, and there was only one other ligand that was regulated by iron in the liver, and that is BMP2. It was decreased by a low iron diet and increased by a high iron diet, although the dynamic range for BMP2 expression was not was less than for BMP6. So these were then attractive candidates for endogenous regulators of hepcidin and iron homeostasis. And so we decided to take a genetic approach in order to determine whether this was in fact the case in vivo. But in order to do this, we needed to understand which cells were responsible for making the BMP ligands, because many BMPs are actually important for embryologic development. In particular, a BMP2 global knockout mice are embryonic lethal, although that's not the case for BMP6. And so we looked at the different cells in the liver, and it turns out that it's the endothelial cells in the liver that are the predominant producers of both BMP6 and BMP2, compared with the parenchymal cells, the hepatocytes, as well as the resident liver macrophages, that is the Kupffer cells. So we then went on to use a genetic approach to look at the effect of knocking out these ligands. And here's data for BMP6. So in red, you can see the global knockout mice. In blue, the endothelial knockout mice. In green, the macrophage-specific knockout mice. And in purple, the hepatocyte-specific knockout mice. And this is, again, for BMP6. And what you can see here is that both global and endothelial BMP6 knockout mice have hepcidin deficiency, whereas there's no effect on hepcidin expression in the macrophage or hepatocyte knockout. And these mice, these both global and endothelial BMP6 knockout mice, also exhibited all of the other features characteristic of hemochromatosis, including massive iron overload in the liver that I'm showing you here. On the left, you see the quantitation, and on the right, the Prussian blue stain, which stains the iron blue. And we saw pretty equivalent levels of iron loading in both the global and the endothelial BMP6 knockout mice. We then went on to show that endothelial BMP2 knockout mice also have hemochromatosis. Here's the data for hepcidin. So it's reduced in both the male and female endothelial BMP2 knockout mice. And again, these mice had all of the features of hemochromatosis, including, as I'm showing you here, massive iron loading in the liver that seemed pretty similar in the endothelial BMP2 and BMP6 knockout mice. And another group in Germany independently also showed a similar findings in endothelial BMP2 knockout mice using a different Cree strain. So together, these data are consistent with this model. This is what I'm showing you here is the liver, and endothelial cells in the liver produce BMP6 and BMP2 ligands, which are secreted and can bind to the co-receptor hemojuvelin on the hepatocyte membrane surface. This acts to enhance the activity of the BMP receptor complex to induce the phosphorylation of SMAD proteins, which is a key transcriptional regulator of hepcidin expression, thereby regulating systemic iron homeostasis. And this pathway is a critical regulator of hepcidin induction by iron. So one of the things that we're focused on in the lab now is trying to better understand how it is that iron, that the signaling pathway is regulated by iron. So we already have some clues as to how this happens, right? From the dietary iron experiment I showed you earlier, where by changing the dietary iron content actually regulates the expression of both BMP6 and BMP2 ligands. And in fact, the expression of BMP6 in particular actually correlates very well with liver iron content, suggesting that it may be the tissue iron content in the liver that is the signal that is regulating BMP6 expression. And in data that I don't have time to go into today, we have shown in collaboration with Hal Drakesmith's group one mechanism by which iron regulates BMP6 production in liver endothelial cells through the NRF2 pathway. And we also now have some new data that we're hoping will be ready to present at some time in the near future, detailing some other mechanisms by which iron regulates the production of the ligands. But there's actually more than one iron signal that regulate hepcidin expression. It's not only tissue iron content in the liver. And that's illustrated by this series of experiments where we treat mice acutely with a dose of iron by oral gavage. And in this experiment, we use a dose of oral iron that leads to a submaximal increase in serum iron levels and transferrin saturation without changing liver iron content. So you can see here that this dose of oral iron is enough to increase transferrin saturation without affecting liver iron. And in this model, this amount of this isolated increase in circulating iron without changing liver iron is sufficient to induce hepcidin expression.
Jodie, can I interrupt you for 1
sure.
second? We have a question in the chat. Iqbal, can you come on and ask?
Yeah, sure.
Great.
Hey, Jodie.
Yeah, hi.
Hey, is there a hierarchy between BMP2 and 6, or clearly the mutants phenocopy each other actually really well?
Yep. So stay tuned. I'm going to talk about that.
All right. Okay.
Good. Thank you.
All right. So these data suggest that, again, serum iron is a distinct or an independent signal to induce hepcidin, and kind of consistent with this idea that changes in liver iron are regulating BMP ligand production. In this gavage model, where liver iron levels don't change, we also don't see any change in BMP6 or BMP2 mRNA levels in this model. However, we do see induction of the downstream SMAD158 signaling cascade. So these data suggest that circulating iron is an independent signal to induce hepcidin, presumably by inducing SMAD signaling, independent or downstream of an upregulation of BMP2 and 6 ligand production, at least at the level of mRNA. And so to think about how circulating iron is regulating hepcidin, let's go back to hemochromatosis, this genetic disorder, and the two other genes that are mutated in hemochromatosis, that is HFE and TFR2. So what do we know about these proteins? So HFE is an MHC class 1-like molecule, and it's been well described through a number of methods, including crystal structure studies, that HFE actually binds to transferrin receptor 1, or TFR1, which is the main receptor responsible for transferrin-bound iron uptake in most cells in the body. And it turns out that the binding site for HFE on TFR1 is actually the same binding site as transferrin, and that iron-bound transferrin or holotransferrin can compete for HFE to bind to TFR1. And transferrin receptor 2 is a homolog of TFR1 with more restricted expression in certain tissues, including the liver. And TFR1 as well can bind directly to holotransferrin. So of course, these proteins are poised to be functioning as important serum iron sensors. And the current working model is that when circulating levels of iron increase, holotransferrin binds to transferrin receptor 1 and competes for HFE binding, which is then displaced. And of course, through the IRP pathway over time, the high iron levels will also reduce TFR1 expression, which will also serve to free up HFE. And Nancy Andrews' group a number of years ago did some really elegant mouse model studies where she made some mutant TFR1 proteins. So she overexpressed a mutant TFR1 that could not bind to transferrin, but could bind to HFE. And she also made a mouse model where she knocked in a mutant TFR1 that could bind to holotransferrin, but could not bind to HFE. And using these mouse models, she showed that HFE, when it's not bound to TFR1, is actually the form of HFE that is signaling to induce hepcidin. And Nancy's group, as well as Carolyn Enns and others, have shown that HFE and transferrin receptor 2 can actually bind directly to each other. And in vitro studies suggest that this binding interaction is important for the induction of hepcidin in response to holotransferrin. And mechanistically, it is thought that the way HFE and TFR2 induce hepcidin is through functional interaction with the BMP-HJV SMAD signaling pathway. And the data for this is based on looking at mouse genetic models and human patients with mutations in these HFE and TFR2, showing that SMAD signaling is defective in the liver of these mice and these patients, coincident with this low hepcidin expression. And in overexpressing models, you get increases in SMAD phosphorylation, which parallels the increases in hepcidin. Mechanistically, there's some in vitro overexpression studies that suggest that TFR2 and HFE can actually bind directly to some components of the BMP-HJV receptor complex, in particular HJV and BMP type 1 receptors. Although whether that is truly the mechanism of action of how HFE and TFR2 actually induce hepcidin, I think there's still some controversy as to whether that's really how it's working in vivo. But nevertheless, this is the current model. So what we've been doing recently in the lab is trying to understand what is the relative role of BMP2 versus BMP6 versus HFE in iron homeostasis regulation. And this is a project that's been led by Xia Xiao in the lab. And to look at this, we utilized a genetic approach where we made different single and double knockout mice to look at the phenotypes to see how these proteins genetically interact. And so one mouse model that we made is we took our endothelial BMP2 knockout mice and we mated them with our endothelial BMP6 knockout mice so that we could generate littermate mice with the same genetic background who either were our sort of control wild type surrogate mice, that in green, mice with a single endothelial BMP2 knockout in red, mice with a single endothelial BMP6 knockout in blue, and mice with a double endothelial BMP2 and 6 knockout. And what we thought we were going to see in this experiment is the following. So historically, when we first identified BMP6 as an important hepcidin regulator, we had recognized at the time that although BMP6 knockout mice have hepcidin deficiency, there is still a residual pathway, a pretty robust pathway for iron to induce hepcidin. And that's illustrated by the following experiment. So if we take these BMP6 knockout mice and we wean them to a low iron diet, and then either keep them on the low iron diet or switch them to a standard diet that has much more iron in it, you can still see a 500-fold induction of hepcidin levels in the BMP6 knockout mice. And these experiments have also been repeated in mice who are on purified diets where the only differences in the diets are iron. So it's really iron that's mediated this effect. And so this was a type of experiment that actually drove us to look for other ligands, and that's how we found BMP2. So when we found BMP2, we said, aha, now we know what's accounting for this residual hepcidin induction by iron in the BMP6 knockout mice. It's going to be BMP2. So our expectation was in the double knockout mice, we would see a more profound hepcidin deficiency, more iron overload, and that we would get rid of this residual induction of hepcidin by iron. So what did we find? So here's the data from the cross. So again, the red is the single endothelial BMP2 knockout mice. The blue is a single endothelial BMP6 knockout mice. And these mice had a similar degree of hepcidin deficiency, about 50 to 60% reduction compared to our controls. So what did we see in the double knockouts? Well, they were actually completely similar to both of the single knockout mice, a similar degree of hepcidin deficiency, a similar degree of iron overload, both in the serum as well as tissues, not only in the liver, but also in extrahepatic tissues. And moreover, when we looked at this residual ability of iron to induce hepcidin in these mice, so here, this is actually a different mouse model where we actually made a global knockout of BMP6 and an endothelial knockout of BMP2, because the endothelial BMP6 knockout were a little bit less severe than the global BMP6. So we wanted to get rid of as much of the BMPs as possible. So this light blue data is single BMP6 knockout mice. And again, as I showed you before, these mice have a residual ability for iron to induce hepcidin. So what happens in the double knockout? This residual hepcidin induction by iron is still there. So together, these data are suggesting that actually BMP2 and BMP6 are predominantly working together to regulate hepcidin and iron homeostasis regulation, and neither one can compensate for the loss of the other. And moreover, there's still some kind of residual pathway, and it's a pretty robust residual pathway for iron to induce hepcidin, even in the absence of both BMP2 and BMP6. So what is this residual pathway? Well, in both the single BMP6 knockout mice on the left and the double BMP6 endothelial BMP2 knockout mice on the right, iron can still induce SMAD signaling, as shown by the phosphorylation of SMAD5 proteins. So this residual pathway appears to be related to SMAD signaling. Moreover, if we repeat this experiment in the BMP6 knockout mice, where we wean them to a low iron diet and then switch them to a regular diet that has more iron in it, if we switch them to a regular diet in combination with a neutralizing BMP antibody that inhibits all of the BMPs that have been shown to regulate hepcidin except BMP9, we completely wipe out this residual hepcidin induction by iron. So these data suggest that whatever the residual signaling is, it must be related to another BMP ligand and the SMAD signaling pathway. Now, what about HFE? What is the relative role of BMP2 and HFE in iron homeostasis regulation? So to ask this question, we mated our endothelial BMP2 knockout mice with global HFE knockout mice, again, to generate littermate mice that have the same genetic background, either with controls, that's the green, a single endothelial BMP2 knockout, the red, global HFE knockout, the yellow, or the double endothelial BMP2 global HFE knockout. So in these mice, what we found, in contrast to the double BMP2, BMP6 knockout mice, in these mice, the double knockout actually did have a much more profound suppression of hepcidin expression. And again, you can see we cut the axis here. So there's a much more profound suppression of hepcidin in the double knockout than either of the single knockout. And this is correlated with much more profound iron overload in the double knockout mice, shown here in the serum as well as the liver. Now, it's important to keep in mind that in these single knockout mice, you already have pretty profound increases in serum iron and liver iron. So the bigger differences are really in extrahepatic iron loading. And in fact, we saw pretty profound iron loading in extrahepatic tissues in these mice already at 8 weeks, both in the pancreas and the heart.
Jodie, we have a
Yeah.
question in the chat.
Sure.
Carol, do you want to ask your question?
Sure. Jodie, in these mouse models, you've got a global BMP6 and an endothelial BMP2, and I understand that that's necessary. Is it possible that there is an extrahepatic source of BMP2 or a non-LSEC source of BMP2 that's giving you this regulation?
So that is a possibility. We can't rule that out because we can't generate obviously global BMP2 knockout because they're embryonic lethal. But this is the best data that we have. So, this is the best way we can look at it. But again, we looked at other tissues and we asked, does iron regulate BMP2 or BMP expression in any other tissues? And it was only in the liver where we saw this regulation, and specifically in the endothelial cells, because the other cells on the liver had very little expression of either of these ligands. So, it's not conclusive proof, but the data suggests that this seems to be the predominant source. But that's the best we could do. Does that answer your question?
To the extent that you're able to answer it, I'm still kind of curious, but yes, thank you.
Jodie, can I piggyback off
Sure.
Caroline and ask you, do you have any data whether BMP2 and 6 heterodimerizes?
So see, you're perfectly setting me up for the next part.
That's going to be $20 more after this.
So stay tuned. This is one of the things I'm going to talk about.
The data is consistent with them synergistically, just based upon the genetics.
Mm-hmm.
That would mean that there must be another BMP when one is being replaced by another in your single knockout. That's how I think about it. But anyway, I'll wait.
This is exactly what we're thinking. So I'll talk to you a little bit about what we're thinking along these lines in a little bit.
Great, thank you.
Sure. Okay, so now again, so what about the residual ability of iron to induce hepcidin in the absence of endothelial BMP2 and BMP6? What happens in these double knockout mice where we lose both endothelial BMP2 and HFE? So here's data in single HFE knockout, and in the single HFE knockout, similar to BMP6 and endothelial BMP2, there's this robust residual pathway for iron to induce hepcidin. But when you knock out HFE, that's completely gone. And as I mentioned briefly earlier, what's the mechanism of action of HFE? It's thought that it's intersecting functionally with the BMP SMAD signaling pathway. And our data are consistent with this because if we look at SMAD signaling in the livers of these mice, as represented by the phosphorylation of SMAD5 proteins, you can see in the circled, red circled images that in the double knockout mice, both in the males and the females, the phosphorylation of SMAD5 protein is significantly reduced in the double HFE endothelial BMP2 knockout mice compared with either of the single knockout mice and also the controls. And that's quantitated over here. And moreover, when we look at the effect of dietary iron, so the switch from the low iron diet to the control diet, whereas in the single knockout mice, there's a residual ability of iron to induce SMAD phosphorylation, similar to the residual ability of iron to induce hepcidin. In the double knockout mice, we completely block this residual ability to induce SMAD phosphorylation. So together, these data suggest, number one, that BMP2 and 6 are working collaboratively to regulate hepcidin expression. And I'll get to Iqbal's comment in a minute, because that's what I'm going to talk about in the last part of the talk. These data also suggest that HFE has the ability to regulate hepcidin independent of BMP2. And in fact, the Roth and Copin group have also shown that HFE has the ability to regulate hepcidin independent of BMP6, which would fit with the idea that BMP2 and 6 are working together. And finally, in addition to BMP2 and BMP6, there's at least one other BMP ligand that contributes a non-redundant role to hepcidin regulation by iron through an HFE-dependent mechanism. And this is another area that we're working on in the lab. And again, hopefully soon, we're hoping to have some data that will be ready to present along these lines. But in the last part of the talk, I want to talk about a new collaborative project that we're working on together with one of your CIHD members at Utah, Jan Christian, where we're trying to understand how it is that BMP2 and 6 are working collaboratively to regulate hepcidin. And of course, as Iqbal mentions, one thing that's important to keep in mind in this field, as I mentioned, BMPs and all TGF-beta superfamily members actually are dimeric proteins. But they not only can form homodimers, where you have two of the same monomers of the same protein coming together, but they can also form heterodimers, where you have monomers from two different BMP ligands that can dimerize. And for the BMPs that regulate hepcidin, in particular, it's been shown that class 1 BMP ligands, BMP2 and 4, can heterodimerize with class 2 BMP ligands, that is BMP 5, 6, and 7. And intriguingly, several groups have shown in the past, this is a publication by another group, that heterodimeric BMP ligands can have actually increased activity over homodimers in some biologic contexts. And here's data in a pre-osteoblast cell line where they looked at the ability of BMPs to induce calcification and mineralization using alizarin red staining and vodkasa staining that's shown here and here respectively. And you can see that the heterodimeric BMP26 protein is more potent to induce calcification and mineralization compared to BMP6 homodimers and BMP2 homodimers, and also compare with no growth factors, and the mineralization percentage is quantified over here. So why would that be? Why might heterodimers have more biologic potency? So one mechanism is suggested by the following set of data from the same paper, where they looked at they used surface plasmon resonance to look at the binding affinities for the different ligands for the BMP receptors. And it turns out there's some differences between class 1 and class 2 BMP ligands. And one of the differences is that class 1 BMP ligands, and here they're showing the data for BMP2, tend to have higher binding affinity for the type 1 receptor. Here they looked at BMP R1A compared to the type 2 receptor. Here they looked at ACTR2B. In contrast, the class 2 BMP, here we're showing the data for BMP6, has higher binding affinity for the class 2 receptor, for the type 2 receptors compared to the type 1 receptors. So you can imagine, and they actually demonstrated it experimentally in this study, that the heterodimer can have higher affinity binding for both the type 1 and the type 2 receptor. So this ability to generate a higher affinity complex may be one mechanism by which these heterodimers can have increased biologic potency. So do these heterodimers really play an important biologic role in vivo? Jan Christian at Utah has published this really elegant study in eLife in 2019, where she showed experimentally that BMP7 actually functions predominantly as a heterodimer with class 1 BMP ligands during mammalian embryogenesis. And I'm going to show you a little bit of data from her paper that demonstrated this. But before I do that, I want to just mention an important facet of BMP biology, and that is that BMPs and all TGF-beta superfamily of proteins are actually made as precursor proteins that are cleaved by proprotein convertases to yield, the mature active signaling ligand as well as the prodomain. So that's an important point to note to understand Jan's study, and I'm going to talk about a little bit about this later as well. So what Jan did in her study is a really sort of, again, really elegant study where she generated a mutant form of BMP7, where she mutated that cleavage site so that it was not able to be cleaved. And the uncleaved protein is not able to signal, okay? And so the hypothesis would be in the study that to look at the phenotype of these animals is that if BMP7 is predominantly acting as a homodimer, these mutant animals would have non-functional BMP7 homodimers. And so they would phenocopy what you would see in BMP7 knockout mice. And again, I don't remember if I mentioned this earlier, but in BMP7 knockout, mice actually live to be born, but then they have perinatal lethality, but they do okay during embryologic development. In contrast, if BMP7 is mainly functioning as a heterodimer, what this non-cleavable mutant will do is it will bind to the class 1 BMP2 and BMP4 proteins. So not only will it, form this non-functional heterodimer, so you lose the activity of BMP7, but because you're sequestering the class 1 BMP ligands and preventing them from potentially interacting with other class 2 ligands, such as BMP5 and BMP6, you can have a dominant negative effect where you start to inhibit the activity of other functions of BMP2 and 4. And we know that BMP2 and 4 are more important during embryogenesis because the global knockout of either of those is embryonic lethal at an early stage. And so the prediction would be if BMP7 is predominantly acting as a heterodimeric ligand, you would have a more severe phenotype in these mutant animals. So what did she find? So these are embryos at day 10.5 and 11.5 from the global BMP7 knockout. This is a mutant BMP7 where she knocked in a tagged functional version of BMP7, and these are wild type. Here's the embryo and here's the heart, and you can see basically a similar phenotype in the global BMP7 knockout as to the wild type and the presence of the flag tag. So what happens with the non-cleavage mutant? Well, they had a more severe phenotype. So you can see here the smaller embryos, which were pretty much resorbate by day E11.5 with significant cardiac abnormalities. So this more severe phenotype is consistent with this idea that BMP7 is predominantly functioning as heterodimer with class 1 and class 2 BMPs. And there was other data supporting this in the paper that I don't have time to go into. And in fact, Jan and her group were able to actually demonstrate the formation of BMP4 and BMP7 heterodimers using co-immunoprecipitation experiments in these embryos. Okay, so this kind of sets the stage to say that, heterodimeric ligands exist, they can have increased potency, and they do have other biological functions. And so, of course, together with our data in the BMP2/6 knockout mice, have us hypothesizing that potentially BMP2/6 heterodimers may be critically important for hepcidin and iron homeostasis regulation. I want to go back and talk a little bit more about this idea of the proteolytic maturation and the protomains of BMP ligands and the importance in BMP ligand function. So this proteolytic maturation actually varies among TGF-beta superfamily members and specifically also the BMP subfamily as far as the number and location of the cleavage sites, whether the prodomain remains associated with the mature ligand after cleavage and other factors. And the protomains are actually critically important to aid in the folding and dimerization of BMP proteins, as well as in regulating ligand stability, function, and in some cases can actually help to target ligands to specific binding partners. And although this has been well studied for some ligands, there's very little known about the maturation and protomains of BMP6 and BMP2. So this is really an area that's ripe for further investigation. But one thing that's been shown for other superfamily members is this interesting finding that I want to show you with an example here. This is pro-activin A. So activin A is another TGF-beta superfamily member. This is the pro protein, and here's the structure. And this is actually a homodimer. You'll see two different colors, but the two different colors are just to show you which of the regions of the protein that are coming from each of the individual monomers. So the green is one monomer and the yellow is the other monomer, although they're both activin A. These GF domains or growth factor domains are that mature ligand domain that's the part of the protein that does the signaling. And these other portions are the prodomains. And you can see that there's interactions between the prodomains and the ligand, the mature ligand domain in this structure. And interestingly, many of the TGF-beta superfamily members have what they call a crossed arm domain swap configuration, where the prodomain of one ligand is actually interacting with the ligand domain of the other monomer. All right, and so if you think about this idea that the prodomains are important for folding of the ligand and for dimerization, the fact that the prodomain binds to the ligand domain of the other monomer raises this idea that prodomains may be very important for determining whether a homodimer is going to be formed or a heterodimer is going to be formed. The importance of prodomains is really illustrated by the fact that point mutations in humans in a number of BMP prodomains have been associated with clinical syndromes. For example, orofacial clefting, microphthalmia, skeletal malformations, and fertility defects. And interestingly, for BMP6, subsequent to our discovery of an important role for BMP6 in hepcidin and iron homeostasis regulation, several groups have now described actually heterozygous mutations in the BMP6 prodomain that lead to inappropriate hepcidin synthesis and iron overload in human patients. And I mentioned that the prodomains and the proteolytic maturation of BMP6 is not well understood, but BMP7 has actually been well studied. And if you line up the sequence that, by sequence homology, BMP6 and BMP7, interestingly, you'll see that where these point mutations that are associated with inappropriate hepcidin synthesis and iron overload exist, two of them are in this green region, which in BMP7 has experimentally been shown to be the ligand interaction domain. So the domain of the that actually binds to the ligand, the active ligand domain of the and so two of the point mutations are located there, and two of them are nearby. And there's another one that's in a conserved region of the prodomain whose function isn't that well understood. So, based on all of this background and these data and what I showed you, we hypothesize that BMP2 heterodimers, yes.
Sorry to interrupt for a second. I can't figure out, there's some feedback coming maybe from your headset. Like, I don't know if that can be draped over your shoulder or something. We're just
sorry, I'm moving my arms around too much. Is this better?
I don't, it's a hypothesis. I don't know, and I know you're almost done, so I'm sorry to interrupt. But
no problem.
Okay.
I will keep my arms still and move this over. Is that better?
I don't know that that's it, but anyway.
Can you hear me better now?
Yes.
Okay. So what we hypothesize, as Iqbal mentioned, is that we think that because of the phenotype of the double knockout mice looks exactly like the phenotype of the single knockout mice, and because of these intriguing data about heterodimers and their importance, their increased biological efficacy and their importance in certain biologic contexts, we hypothesize that BMP26 heterodimers may be the key functional ligand for hepcidin and iron homeostasis regulation. And moreover, we hypothesize that the prodomain of BMP6 and also possibly BMP2 may play a critical role in BMP26 heterodimer formation and function in regards to hepcidin and iron homeostasis. So Jan and I have teamed up, and we're now thankfully funded through the NIDDK to further explore these hypotheses. And so currently we're working on 3 aims. One is to characterize the proteolytic maturation of BMP6 and BMP2 homodimers and the BMP26 heterodimer. We're going to test whether the prodomain of BMP6 and or BMP2 is required for homodimer and or heterodimer formation and function and hepcidin regulation. And we're going to explore the impact of these BMP6 prodomain mutations that were associated with iron overload in humans on BMP26 heterodimer and also homodimer formation and function in regards to hepcidin and iron homeostasis regulation. So we're really excited and, we don't have data ready to show you yet on this, but hopefully if you invite us back in the future, we can show you some of our progress along these lines. And I think this holds the promise of really better understanding the mechanisms of hepcidin regulation and also providing more insights into BMP biology in general. So finally, I'll thank by acknowledging the folks in my lab who have contributed to the data that I showed you. And of course, give my thanks to Jan, who we're now collaborating with, and we're looking forward to further work on this project. I want to also acknowledge my funding sources and thank you for your attention.
Thank you so much, Jodie. Great presentation. Super interesting to think about that subtle regulation by the homo-heterodimer formation. And whether, I was trying to figure out in some of the animals you showed, when you gave them iron, they had these really huge induction. It kind of looked like outlier sets. And maybe those have different levels of some of these other BMPs that are regulating that piece, because I'm always trying to figure out what those outliers have that's different, that's making them so responsive to the iron. We have a question in the chat. John, do you want to go ahead and ask it? John Phillips.
Hi, Jodie. Hey,
Hi.
if one only expresses just the
Hey, John, you got to turn on your video to ask a question.
He could be in his jammies. Oh, nope. Okay.
No, I'm here in my office. If one just expresses just the functional domain, not the whole thing with the prodomain, can you gain any sort of insight into whether there's true function for that prodomain by just, if you only express the functional domain, can you bypass the lethality, I guess, of the double knockout?
So, I mean, I think that you're not going to get, these prodomains are important to generate the for the proper folding and formation of these proteins. So, I think that experiment may be challenging to do, but this is there's not that much known about it. So these are the types of experiments that we're doing to kind of understand, are the prodomains necessary to form the functional dimeric proteins, either homodimers or heterodimers, and which prodomains are important and what are the impacts of the different regions in the prodomains for the function. I mean, certainly the signaling experiments that I showed you with recombinant protein, these are just the intact, the mature ligands that are doing the signaling, but how they're made and how the endogenous proteins, how they're made and what are the impacts on their regulation and function. There's really not much known about that.
Can I ask a question, Diane?
Yeah, go for it.
So Jodie, the way I thought about it is, I've been thinking about this from our own work being dragged into BMPs and C. elegans. So perhaps what's happening is a graded response. It's different versus low versus high iron. It's a graded response where you have BMP6, BMP6, BMP2, BMP2, BMP4, BMP6, and they form multimerization or heterodimerization or whatever you want to call it to be able to provide graded responses because they're very different dissociation constants, which allows fine tuning of iron metabolism because you don't go from low to super high iron concentrations, right? Everything is a shade of gray. And that is what your phenotype is complex. And perhaps the prodomain mutations will allow you to be able to identify how many different components BMP6 or 2 interacts with.
Yeah, I think that the fact that these two ligands were the only ones that are regulated by iron, and particularly induced by a high iron diet, because a lot of the, I think that
of iron by iron may not have anything to do with quantities of BMPs that's found to be able to interact with your main effector molecules. So it's essentially you may have accelerators being turned on, but the brakes might already be there. That's what I'm trying to get to.
Yeah, definitely. I think all of these things are possibilities. And I think that I mean, these particular ligands, I think are important under high iron conditions, but under sort of normal conditions or lower iron conditions, I think that's where you have many of these other regulators. So we'll have to see, but it was really, to me, surprising that you couldn't have some compensation in the double knockout mice, right? You're still going to be able to make BMP2 homodimers, and BMP2 is still there in the BMP6 knockout mice. So why isn't there any function in those, any difference in the phenotype in those animals, right? So if BMP2 homodimers were doing something, or BMP2 was able to heterodimerize with something else, you would think that once you knocked out BMP2 in addition to BMP6, you would see some difference in the phenotype, but there was nothing. So to me, that's really a fascinating question, because BMP2 is still there in the BMP6 knockout mice. Why doesn't it do anything? So this is what we don't understand.
I have a question.
Go ahead, Caroline.
So at one point, it seemed like the thinking was that the BMP6 responded to sort of a physiologic range of dietary iron, and that BMP2 kicked in at this supraphysiologic range. But that doesn't sound like it's coming out of the data you have now anymore. You don't think that?
No.
No.
They only seem to be working together, because again, if there was some function of one without the other at a lower iron range, then you would expect to see some worsening of the phenotype when you took out the other ligand. When one ligand was still there, if it was doing anything, and it's not. So that was, again, particularly surprising to me, because I was not expecting that. There still may have been some other role for other ligands, but I was expecting to see some difference between the single knockout. But
so the other question that I have is, and I'm assuming that because you haven't talked about it, that the answer is no, but have you found any, GWAS data or human mutations or, different alleles in the human population for any of these proteins that might give you some hints as to, in humans, what's really happening?
Yeah, so there have been some studies that have suggested that there might be some variations in BMP2 or BMP4 that might be modifiers of iron in hemochromatosis, but really there's not a lot of signal there. And there's different reasons for this. One could be that those ligands are so important for other things that if you have a problem with the signaling, that you have too many other problems, and so they don't exist. Development, right. And so, I mean, that's one possibility. But I mean, all we know is what we see out there. So we haven't really gotten good clues from that to clue us in as to which ones are important. But what I don't understand, again, is why wouldn't a homodimer form and be able to signal, right? If BMP6 is not there, BMP2 is still there. Why doesn't it do anything, and vice versa? Why doesn't does the homodimer not able to form, or is it just not functional? Why is that? This is a mystery to me that I'm still trying to figure out.
Is there any structural data that would be informing on that?
Which was the next question I was going to ask you. Did you use Google's DeepMind to model?
I have not done that, no.
Oh, it works really beautifully. You should really consider, especially since I can't remember what you showed, but you showed differences in the pro domain between BMP6 and BMP7,
Yeah,
correct?
the
So-
prodomains, the prodomains are definitely have differences, yes, and they're
Right.
big.
So, and you
yeah.
said that, one prodomain binds into the ligand binding pocket of the other. So I think that's going to be fun for you to use DeepMind to get structural information between BMP6 and BMP2 and BMP7, force it with BMP2 or any of the other guys
Yeah.
to see whether it works or not.
Yeah, this is a whole sort of unexplored area, so there's a lot we can do.
Cool. Any other questions? Jodie, great talk. Thank you so much. Thanks for all the questions and the interest. And if you have any other comments or questions, feel free to reach out to Jodie. And I want to thank you all for your attendance. And we have one more of these scheduled for this year, which is Stefano Ravella. And so watch for the flyers coming out to let you know about that. And thanks, you guys. And don't forget to check out the websites for funding opportunities and core facility information. We've got some really great cores going on right now that are new and exciting. At the CIHD, we have a protein metabolite core that's just come online, which is really taking us to another level of, I think John
Omics.
used the word omics, yeah, where you can now see how metabolites are going to regulate your protein of interest. So I highly recommend you go check that out. So again, thank you, Jodie, so much. And thanks for everybody's attendance, and hope you all stay safe.
Thank you, Jodie.
Everybody, it was good to see you.
Nice
Bye.
to see you.